All right, so now let's talk about blood. So blood. Okay, so I will skip all this stuff, right? So you should already know all this stuff from anatomy. Okay, so we'll start with plasma. Okay, so fluid part of blood. Okay, so again, these are really basic, simple, straightforward stuff. Okay. Okay, so the plasma proteins, okay, makes up 7 to 8% plasma. Albumin, okay, the purpose of albumin is it creates the osmotic pressure to help draw water from the tissues into the capillaries and help maintain blood volume and pressure. Okay, so what happens is the osmotic pressure of albumin causes fluid to leak into the capillaries. Okay, so without albumin, what blood will do is leak out of the capillaries, okay, and end up in the extracellular fluid. Okay, so you see this with kwashiorkor. It's the little starving African children that you see, like with like full bellies and stuff like that. That's not because their bellies are actually full; they're filled with the fluid that's leaking out of their blood vessels. Their diets are so protein poor that they don't have enough albumin. Okay. Globulins, the alpha and beta globulins transport lipids and fat soluble vitamins. The gamma globulins are the immunity stuff that will learn in the immune system. Okay, fibrinogen, okay, is a water soluble protein, but it can become cleaved into fibrin. Okay, and serum by definition is the blood without fibrinogen, so serum can't clot. Okay, the plasma volume. Okay, the regulatory mechanism maintain plasma volume to maintain blood pressure. The osmoreceptors in the hypothalamus, okay, cause the release of ADH if there's too little fluid. Okay. All right. So erythrocytes, I'm sure you guys know this. Anemia is abnormally low hemoglobin count or red blood cell count. Okay. So here's 3 types of anemia. You guys know that. Okay. Okay. So white blood cells. Okay. Have nuclei and mitochondria. They move like amoebas. Okay, so amoeboid fashion. Okay. So the types of leukocytes, granular leukocytes, okay, they have granules in them. That's why they're called granular leukocytes. Neutrophils, eosinophils, basophils, agranular leukocytes, monocytes, and lymphocytes. Like I taught you guys in micro, never let monkeys eat bananas. Okay. So never let monkeys eat bananas. And that's the mnemonic from most to least. Okay. Okay, platelets or thrombocytes, okay, are actually not cells, despite the name. They're actually fragments of a larger cell called megakaryocyte. Okay, they're little pieces. They're short-lived. What they do is they clot blood along with fibrinogen. Okay. All right, and here's what they do, right? So here's the functions. okay, of the different cells. Okay. One second. All right. Couple of things. Hematopoietic stem cells. So these things that form blood, they're hematopoietic stem cells or hemopoiesis. Okay. This occurs in red bone marrow. Okay, and the cells differentiate. They develop memory receptors for different signals and processing. Okay. So first one, erythropoiesis. Okay. Formation of red blood cells. Okay. Your red bone marrow produces about 2.5 million red blood cells per second. So think about how much that actually is. And that's also how much you lose every second. Okay. Regulation of this. This is stimulated by EPO, erythropoietin. Okay. Your kidneys measure the amount of red blood cells coming through, and they respond to EPO, okay? All right, one of the major components of red blood cells is iron, okay? So most of the iron is recycled, but you always need a little bit of iron coming from the diet to maintain it, okay? Without iron, you get anemia, okay? Most of the iron is secreted into the blood through thermopoietin channels. Okay, iron does not travel freely in the blood. Iron is something that you actually have to keep very tight control over because bacteria love iron, and so we have sequestered the iron away from bacteria. Okay, so all iron travels in blood by the transferrin. Okay. Okay. So, leukopoiesis. Okay. Depends on interleukins and other growth factors. Okay. Thrombolysis. Okay. Is basically the breaking off parts of the megakaryocytes. Okay. Thrombocytosis is abnormally high platelet count. Okay. Thrombopenia is low platelet count. Okay. Red blood cell antigens and blood typing. This I'm pretty sure everybody understands. Okay, so I will the label that you basically have to match antibody types and stuff. Like if you actually don't understand, just let me know. Okay, and this augmentation is equal blood typing. Okay, Rh factor is antigen D. Most people are Rh positive. About 85% of people are Rh positive, which means they have the antigen. Okay, Rh negative does not have the antigen and does not have antibodies to the antigen, and that is a problem. Okay, unless exposed to Rh positive, either through blood transfusion or pregnancy. Okay, issues in pregnancy. A Rh mother exposed to Rh positive blood produces antibodies. Okay, this will affect not the first pregnancy because the first pregnancy, you're not exposed to the fetal blood until childbirth. The second pregnancy might have a problem because you're already exposed, so you have antibodies to Rh factor. Those antibodies can cross the placenta and attack the fetus, okay, which will result in urethral blood stenosis fetalis. Okay, to prevent this, the Rh mother, the Rh minus mother, is treated with RhoGAM. RhoGAM is basically an antibody that attacks Rh factor by itself, so the mother has no memory of it. Okay. All right. Hemostasis is to prevent bleeding. Okay. So damage to collagen fibers in the blood produces vasoconstriction, so less blood flow through, formation of the platelet. That's the initial plug, and then eventually that gives way to a fibrin protein web. Okay. Okay. So normally your blood flows through. Okay. The intact endothelium constantly produces nitric oxide and prostacyclin, which causes vasodilation of the capillary and prevents platelet aggregation. Okay, the last thing you want to do is have a bunch of platelets together, sticking together, because they trigger other stuff. Okay, so having platelets together will trigger clots. So last thing you want to do. Okay. Okay, when you damage the wall, okay, when you damage the endothelium, it exposes the collagen. Okay, the platelets bind to collagen, the von Hildebrand factor holds them there, okay, which attracts more platelets. So this is a positive feedback response. Platelets recruit more platelets, okay, which ultimately causes clotting. Okay, so the platelet release reaction, blah, blah, ADP, serotonin, which causes vasoconstriction, and thromboexin A. Okay, so there is a clot. Okay. All right, so the clot starts to form, but it needs fibrin. Fibrin are the mesh. See the big mesh that traps not only the mesh, but there's a whole bunch of red blood cells that trap within that mesh, and platelets trap within that mesh, and like that whole thing is your clot. Okay. Okay. Fibrin is converted to, or fibrinogen is converted to fibrin in one of two ways. Okay, so let's talk about the common path first. Okay, ultimately, they both end up in this common pathway. Okay, clotting factor X is activated. Okay, clotting factor X is activated. Factor 10. I don't know why I call it factor X. Factor 10 is activated, which cleaves prothrombin into thrombin. Thrombin then cleaves fibrinogen into fibrin. Okay. Ultimately, you get a fibrin polymer. Okay, which goes from soluble to insoluble right away. So it goes from soluble to insoluble right at a point where you need the fibers. Okay, so here's the intrinsic pathway, here's the extrinsic pathway. Or, wow, extrinsic pathway in blue, intrinsic pathway in the yellow. Okay. All right. Here you can see the hemophilia and other clotting things. Okay. Okay, and other things we take, coumadin, etc., etc. Okay. Okay. Clotting factors. Okay. So after you finish clotting, the solution of clots plasmin, digest fibrin. Okay. Clotting can be prevented with certain drugs. So calcium chelators, heparin blocks thrombin, okay, Coumadin, or also known as warfarin, inhibits vitamin K. Vitamin K is necessary to a lot of this response right here. Okay. 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 The four chambers of the heart, I'm sure you guys know already. The fibrous skeleton, which I'm sure you guys already know. Okay. Okay, so heart cells. Okay, so all that is anatomy. You can look at anatomy chapters or the recordings, etc. Okay. Okay, heart murmurs. Okay. Heart murmurs are abnormal heart sounds producer blood flow. Okay. Mitral stenosis is the mitral valve calcifies and impairs flow between the left atrium and ventricle, and it results in pulmonary hypertension. Okay, so let me explain why that is. Okay, if the mitral valve calcifies, then it's hard to get from the the left ventricle atrium to the left ventricle, so the blood stays there and it backs up, into the pulmonary circulation, which is why you get pulmonary hypertension. Okay. Okay. In competent valves or where they don't close properly, or even prolapse. Prolapse means it instead of the mitral and tricuspid valve usually sit like this when they close, and when the pressure is on, it flips closed like this and stays there. But if you can blow it open, it prolapses. Okay. Okay. Septal defects, a hole either in the interventricular septum, okay, VSD, okay, or ASD. Okay. Ventricular septal defect or atrial septal defect. Okay. And the most common one is ventricle. Okay. Okay. So here's a ventricular defect. Okay, remember the left side of the heart is bigger, right, and stronger. So it will literally shunt some of the blood into the right side. And so instead of going all into the aorta. Okay. Okay, this is a septal defect in the aorta. Okay, this is the septal defect in the ventricles. Okay, the cardiac cycle. Okay, first of all, systole is a contraction of the heart muscles, diastole is the relaxation. Okay. Okay, end diastolic volume is the total, or EDV is the total volume of the blood in the ventricles at the end of diastole. Okay, and systolic volume is the total amount of blood left in the ventricle after systole. Okay, and it's usually 1/3 of the end diastolic volume. Okay, so your heart doesn't squeeze all the blood out; it squeezes 2/3 of it out. Okay, at rest. Okay. So pressure changes during the cardiac cycle. Okay, the ventricles begin contraction, the pressure rises, and the AV valves close. Okay, and that causes the isoval limit contraction. The pressure builds, the semilunar valve open. Okay, and the blood is injected into the capillary. So these valves, the AV valves close, and then it forces the semilunar valves open and blood is injected in the capillaries. Okay, after the injection, the pressure in the ventricles falls. The semilunar valves close, so you can't have any blood coming back in. Okay, and then the ventricle relaxes. Okay. Okay. So there's a pressure drop in the ventricles, and then the ventricle pressure drops below that of the atria, and that causes the AV valve to flip open, and the blood goes from the atria to the ventricles. Okay, and then finally the atria contracts, sending the last of the blood to the ventricles. The atria contraction sends about something like 15–20% to the ventricles. It's not like a critical thing, actually. Okay. Okay. All right, so, okay. Electrical activity of the heart. Okay, first of all, I explained this a little bit when we talked about muscles, but the heart is autorhythmic. Okay, the pacemaker of the heart is the SA node. The SA node is located in the right atrium. Okay, okay, so the SA node generates something called sinus rhythm. Okay, the AV node and Purkinje fibers are secondary pacemakers of the atomic pacemakers. They beat at a slower rate than sinus rhythm. Okay, but we usually don't allow that. Usually the AV node, listen to the SA node, the pacemaker of the heart. Okay. Okay. So the pacemaker sets itself off, okay, by a slow, spontaneous depolarization. Okay. Okay. What we do actually is we slow down or speed up the node. Okay. Speeding up. Okay. Slowing down. Okay. Okay. So again, the contractions are fairly straightforward. Okay. But there's actually a long plateau that prevents summation. So you can't it's not like skeletal muscle where you can add up pulses. There's a relaxation that is longer than a beat. So the muscle has time to relax. So you can't build up signals. There's no summation here. Okay. Okay. So you need to go all the way out before you can beat again. Those 250 milliseconds really limits the speed of your heartbeat. All right. So, okay, here's the SA node. Okay, the SA node causes the atria to contract. The atria contract independently of the ventricles. Okay, the atria contract together, they have one skeleton. The ventricles contract together, they have another skeleton. Okay, so this contracts, the signal gets sent to the AV node, but there's a brief 100 millisecond stoppage, and then the signal gets passed down through the bundle of His, okay, through the left and right bundle branches to the Purkinje fibers, okay, and then the ventricles contract. Okay, if you inactivate the SA node, the AV node takes over, but the AV node has a heartbeat rate of about 50, okay? Okay, so there is a conduction of impulses, etc., etc. So, lub dub. The lub dub is atria, the split second weight, then ventricles. Okay. Calcium release, etc. So there's an absolute refractory period before you can start off again. Okay. Okay. The electrocardiograph records the activity of the heart by picking up the movement of ions. Okay. Okay. So the electrocardiogram waves and intervals. So P wave. Okay. So P wave. Okay. QRS complex, T wave. Okay. Or QRS wave. Usually complex is the other term. Okay. The PQ interval is the atrial systole. Okay. The ST segment is the ventricular systole. Okay. And then the T wave is ventricular repolarization. Okay. 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 So atria contract, ventricles contract. Okay. So you can see the different the relationship between the impulse conduction. So sorry, this is the P wave right here. This will cause a ventricular contraction. Okay, and then so that causes depolarization. Here it goes to the AV node that causes the ventricular depolarization, and then finally the repolarization. Okay. All right, so you look at the leads where you hook them up to read these. Okay. Okay. Oh, the ECG. Okay, and heart sounds. The love occurs after the QRS wave. Okay, as the AV valves close. The dub occurs at the beginning of T wave when the SL valves close, semilunar valves close. Okay. Okay. Arrhythmias. So arrhythmias are abnormal patterns of electrical activity that results in abnormalities of the heartbeat. Okay. So they're the 4 drugs. Okay. And you read about what they do here. Okay. All right, I'm gonna cover the rest of this in part 2 of this chapter, okay? Because this chapter is pretty long. All right, so